Okay, well, let's get going. So this is the December ECSS Symposium. I'm Sergio Sanjulevich, Deputy Director of ECSS. And welcome, and we very much appreciate your presence today. It's going to be a great talk. As you can see, it's a very interesting topic. And so without further ado, I will hand it over to Rob. And so I guess, Rob, would you like to people to put questions in chat and we all address them at the end, or how would you like to do that?
If it's stayed a group of, it's 11. I would be perfectly happy if people want to ask questions as we go, or we can wait, or they can wait till the end if they'd rather do that. But I'm perfectly happy being interrupted if you have a question.
Well, yeah, actually, I would say let's put the questions in chat and let's give Rob and Johnny, the chance to expound what they want to say, and then hopefully we'll have like 20 minutes at the end where we can ask all the questions. So I would ask, if you have questions, just drop them in the chat and we'll address them after Rob and Johnny are done. Thank you. So with that, Rob, please take it away.
Sure. Thanks, Sergio. And good afternoon to those of you on the in the east part of the country, and good morning to those in the west. I'm going to talk in this symposium about Alzheimer's disease and some drug discovery projects that have popped up here at the IU School of Medicine in collaboration with some other universities, and I'll discuss that at length. But I want to start by saying I've been involved with academic research computing for almost 20 years now, and have provided a lot of effort to a lot of projects. I cut my teeth on the CERN projects of Atlas and CMS. I've been involved with LIGO, where there's been very massively important discoveries. But I think few projects that I've worked with have the potential for immediate real-world impact, like the one that we're going to talk about today. And this one is a drug discovery project centering around Alzheimer's disease. And in fact, to get a few of the acronyms out of the way here on my initial slide, TREATAD, TREAT is the target enablement to accelerate therapy development for Alzheimer's disease. And the AD Explorer, obviously AD is Alzheimer's disease, and we use that one repeatedly through here. But it is a collaborative project. It's funded by the National Science Foundation. There are two arms of this. One is led by Emory with Sage Bio Networks, and the other one is a partnership between the IU School of Medicine and Purdue University. You can see many of the names of the people who work on the project here at the IU School of Medicine or within IU. There are some names that I'm sure you recognize on this top line, and they're at the IU Department of Biohealth Informatics. Johnny was nice enough to join us. His name there, and Johnny, I'm sorry if I mispronounce this, but Jiannan Lu is his name. He goes by Johnny, and he'll talk to us about the AD Explorer here in just a few slides. So let's see if I can move my slides forward. Okay, so first, a brief note about Alzheimer's disease itself. It is a progressive age-related neurodegenerative disorder. Adults 65 and over, and this comes from the National Institute on Aging, adults 65 and over, about 1 in 10 develop Alzheimer's disease, develop dementia from Alzheimer's disease. About 1 in 3 adults over 85 develop this, and you can see that the CDC reports many deaths. In 2017 was the last time I could get numbers. So I like having about 10 people in the room, because that makes looks like we're up to 15, makes the numbers easy. Of the 14 people here, about 1.5 of us would expect to be impacted by Alzheimer's disease. Once we hit 85, 1 in 5 of us as we age to 85 and over. There is, I can assume that we have a pretty educated group here today, and there is less increase or decreased risk over time for based on educational completion. So maybe those numbers are a little bit high, but I think it's safe to say that many of us will be impacted either directly or indirectly by Alzheimer's disease. Now, and it is really a heartbreaking disease. It destroys memory skills, thinking skills, and eventually the ability to carry out very basic daily activities, often leading to death from malnutrition or dehydration as the basic skills of even eating and drinking are lost. So what is the IU School of Medicine doing about it? And the IU School of Medicine, in partnership with Purdue, they have this thing called the TREATAD Center that really has 4 arms. Those include a computational biology group, and that's the group that we're most heavily involved with, but also a structural biology group, a high throughput screening group, and a med chem group. Each of these working on doing preclinical target validation. TaRget is looking at various drugs and the properties of various drugs and determining if they're a promising target to then move on from a more preclinical stage into a clinical stage of development. And really they're looking for lead molecules and creating a target package for researchers to then down the road expand upon. So this was the initial idea, and I'll talk about how this has changed between year 1 and year 2, which we're in now. But the initial idea was to pull these groups together. You'll notice that the green things here are the various different groups, experimental groups and simulation groups that are working. They're then interacting with a vendor database, which I'll talk about a little bit. The blue dot over here is the BCB portal, where we're trying to deliver additional tools outside of those groups. And those are many bioinformatics researchers here at the IU School of Medicine and in Purdue, and then present the data through an open access data system, or OADS. Now, this is something that has changed between year 1 and year 2. And CIRC, which is the Cyber Infrastructure Integration Research Center, which myself, Marlon, Suresh, Aroma, the people within the IU Gateways group are developing. The initial idea was to develop the BCB portal and the open access data system, but this has changed over time, and I'll talk about how that's changed over time. Synapse.org is a delivery system for AD information, and what we really were trying to produce are these target enablement packages, which really detail the properties of a drug, as I said, as that drug has been researched preclinically and moving on into a clinical situation. So the BCB portal itself, and I'll show this in a little bit, the goals for that were to design and run the infrastructure for the TREATAD IU School of Medicine Purdue collaboration. There's a different infrastructure for the Emory Sage BioNetworks collaboration. And they're looking for new methods of predicting and prioritizing drugs. And really what CIRC is doing throughout this is providing IT support for these bioinformatic projects many are developed by the researchers, many by grad students working with the researchers, but we want to preserve this in a public portal so the people who are using these tools or can benefit from these tools have a place, a gateway to come and research them. The core components consist of several things, some analysis software or tools created by the researchers, grad students, some tools that are just open source tools that are nice to package within a gateway environment for researchers. There are there is mostly openly available data, and we are working with some clinical data, but that's yet to be interacted with. Most of this data that is being interacted with are public sources, including the AMP-AD, PubMed, the Gene Expression Omnibus, and the Chemical Library. And these are openly available. The computing uses some Exceed resources and some local resources here at Indiana University. And some of the persistent services are hosted within the Jetstream cloud environment. We do use Apache Ariavada for delivery of the joint portal. When we pull these things under a single umbrella, some of the tools right now are living outside of Apache Ariavada. We will pull those together over time and make this a single gateway. And then we are looking at how that data management and sharing should take place, what can be published, how it should be published, what license it should be under, what technology it should be working with, and things along those lines. This changed between year 1 and year 2, so we're still now adjusting to the change based on requests from the NIH program officers. So there's a diverse analysis software here, topics, Tsunami, both of which I'll show briefly and talk about later. I didn't put the full name of Tsunami here. I see I missed that, but there are other AD things, including the next to last bullet is the AD Explorer, which Johnny will talk about after I finish up with this slide. And then there are additional projects to be pulled together. So the general idea is to pull all these tools, the data, and things together behind a single gateway where researchers can find a variety of AD analysis tools. So with that, I will turn it over to Johnny. He's been the primary developer of the AD Explorer, and he's going to do a demo. If you want to follow along with the AD Explorer, it's available at adexplorer.medicine.iu.edu. You can follow along there, or just listen and visit that later. I'll make these slides available with the presentation, so you can click on that, as well as some other tools that I'll talk about a bit later. So Johnny, I'm going to stop my share.
Okay.
And let you have it.
Yeah, let me share my screen. Yeah, can you see my screen?
Yes.
Okay, nice. Thanks, Rob, for the introduction of the project, the AD Explorer project. So today I'm just going quickly through the other project we have already developed. I think we finalized this kind of project. So this is a version 1 of the AD Explorer. It's already finalized. So we have 5 modules. Yeah, just quickly show. So here is the homepage of the website. So our tool is focusing on the AD research or neurodegenerative disease. So basically this tool is trying to provide bioinformatics support, focusing on the whole research life cycle of AD researchers. So trying to provide all sorts of resources and tools that AD researchers can use to help with their research. Here is a website map of our ADE. You can see there are 5 main modules here. Each of them focus on different areas of the bioinformatics. And I'm just going to through each of the modules one by one, because you see these 5 modules are arranged in a certain way, focusing on the whole research life cycle. So the first step of the research is usually the literature review. So what we did in the PubAD is trying to collect the publication information from the PubMed database and then process it. We do have an in-house NLP pipeline, the natural language processing pipeline, to analyze all the literatures which mention the AD or related neurodegenerative disease, and then we categorize them and process them to extract the useful information in there, and then sort them with a whole gene list. We have about 20,000 genes, so that the user can query each gene in our like a whole gene list to see the publication information related to a certain gene. So for example, a very common gene in the AD research area is like APOE. If you query one gene, so there is a result page we generated with various visualizations. So first part is basic gene information, very straightforward. And then we have a bar plot for the number of publications in each year. All these publications recorded here mentioned this specific APOE gene. When we process the publication data, we pay a special attention to the alias or different like names of the genes, so that we make it as accurate as possible to have a like accurate description of the publications regarding this gene in the AD research area. When we process the data, we have multiple neurodegenerative diseases, not only the AD, not only the Alzheimer's disease, but we are having like other kind of neurodegenerative disease. Sorry about that. So with our in-house natural language processing pipeline, we have different categories of the data here. First one is the keywords. So those keywords are summarized by an expert in the AD research area. So they are just providing me some very important hypotheses, compounds, or some like tissue types they are very interested in, or there are some very significant importance in the research area, in the AD research area. So we just extract this kind of information from the publications, then try to record the PMIDs so that we can see which keyword has the most publications. And also, just another important point is like for each of the bar here, all the publications in this like 251 publications, all those publications mentioned APOE in it. Also with this kind of realization, we make it make all the bars clickable. So if you click one bar, you can go to the PubMed website to check all those like 251 publications directly, to check the details of those publications. So all basically all bar charts in this page are clickable, so that you can directly click and see the original publications on the PubMed database. And we have the dimension types, as I mentioned. So we are not only including the Alzheimer's disease information. I just saw one question in the chat, so I just quickly answer that. The NLP software, we're using NLTK. It's a Python package, because we use several like different softwares for the publication collection. We use bio Python package. There is a API to communicate with PubMed. Then we extract the data and process and use LTK Python package to process the whole information we collected from the PubMed. And then the final format of the information we shown here is CSV file. It's a huge CSV file. We collected the PMIDs together with different categories of data and store it in the database. So that's what we did for the data processing part. For the detailed processing, so we are preparing a paper for the data processing process here. I think it should be published next year, early next year. Yeah, so here is dimension types and the brain regions, because different researchers may focus on certain brain regions. They want to check the literatures regarding a certain brain region. And also the mouse models, which is very important in the AD. So because people will do experiments and they want to check what kind of publications available for the mouse models they are interested in. And also co-occurred genes. This is basically because we only process the title and abstract of the publications. So when two genes are occurred simultaneously in one publication, which is recorded, and it's kind of the linkage between two genes we inferred from publications. So we can just plot here and see from publications which gene mentioned mostly with the query gene, which is APOE here. And also here we integrated some realizations from the CTD database. This is a chemical database. It's like whether the disease has some relationships or inference. Like there are some relationship inference from the publication, also publications, and then we record it here. So the higher the score, the gene, disease, and chemical relationship is stronger. But we borrow this data from the CTD database and make these kind of realizations for different disease, so that if people are interested in some impacts of the environmental chemical, like some moleculars or some chemical stuff, they can check here whether they have some publications available out there, like how the copper will impact the Alzheimer's disease and together with the APOEG. So you can just check the publications on the PubMed database. So basically the first part is focusing on the publication information. And then the second one, we do have a, so I will mention it in the like because we have a documentation page so that you can check, like for the PubAD, we do have a short description of what we have used and what are all the functions available here. So you can check the documentations for right now. But I think the data processing part will be published shortly. The gene AD part is trying to provide some bioinformatics results, some bioinformatics analysis results, or some collective information we collected from multiple publications, and trying to integrate them together and provide a portal to for the users to query. Like again, for the EPOE gene, we have like multiple tabs in the result page. So the basic gene information here, and differentially expressed gene results. So we don't have any data, but I will just try another one. Maybe this one is not good one for demonstration here. So for the STAT3, which is a very important transcriptional factor in the genome. So for the D results, it basically means differentially expressed gene results. So you can see there are many, each row here is a data set, and this gene is differentially expressed between the AD patients and normal patients in these data sets. So these are data set names. And if you want to check the gene expression values, like gene expression levels of STAT3 in a certain data set, you can just click the zoom and then you can see a box plot for this data set and this gene. So this is 80 patients expression level of STAT3, and this is a non-80 patients STAT3 expression level. So you can check it directly. Try to see how big the difference is in there. And also the gene modules. Those information are from, some of them are from publications. We have the PRMIDs here, collected all those information. And some of them are from our own algorithm. So in our lab, we do have the gene co-expression mining, gene co-expression module mining algorithm, so that we mine the data sites and try to curate or annotate those kind of gene co-expression modules. And then we will incorporate those results here. And the cell markers, so we don't have information for STAT3, but we have some information for some other genes. So whether this gene can be a potential cell marker for a certain cell type in the brain. In the brain, there are lots of different cell types. So it's very complex. So there's some researchers are interested in whether this gene can be a potential marker for a certain cell type, so that they can check this information here. And also the single cell analyze results. Basically, the gene expression, differentially expressed genes in the single cell data sets. So you can see for different cell types, STAT3 is differentially expressed under different conditions. You can check those kind of like analyze results here. We integrate here. We also have the multiple gene query here. Let me quickly get one gene list. So suppose this is like a testing random gene list. And if you query multiple genes, we provide similar information, but there is something different. Yeah, it takes several seconds here. So all the genes information here. In the gene models, this is a different part. Because when you query multiple genes, you want to try to see the overlaps between the query gene set and the gene models in our system, so that we do a feature exact test to see the overlaps, whether there is a significant overlap between the query gene set, like 8 genes, with all the available gene models in our system, so that you can see your gene set is like largely overlapped with this one, because our gene set query is very small. So it's like doesn't mean it's related to this gene model, but it's some information provided here. And also we have annotations or descriptions of the gene model, so that you can see whether your gene list has some significant mechanism or significant functions there. No, so we are just finalize this project right now. I think in the last 1 or 2 weeks. So we're not published like we're not make it like a public available for a long time. We just finalized this project weeks ago. So it's like still pretty new. So this tool is pretty new, but we're trying to make it public available right now. And the next part is the Toolbox AD. So in this, okay, I saw several questions here. So yeah, what they are practitioners, I'm not sure what you mean by practitioner, because we, in our team we have, so this tool is primarily used by the AD researchers. I mean, those kind of experimental researchers who do experiments, or I should say a bench scientist who do experiments.
Yeah, so I mean, that's the kind of practitioners I meant. That is like, you know, it's yeah, it's pretty much obvious, but you know, the questions to ask or what would be important and how this all is laid out must have been informed by people who are actually doing research in these fields, like by what they want to see, right?
Yeah,
how they want to see it.
Yeah, so we have like, because in our team we have like many professors working on the AD research. So during the development process, I'm talking a lot with them, and they are providing lots of different feedbacks, so that we want to make our tool to be user friendly as much as possible. Right. So it's like just trying to. So
they're in the loop very actively.
Yes. Yeah.
Sergio, additionally, we've worked with TREATAD, though not necessarily this tool, but with many of the tools with our user experience experts. So I'll talk about the CDD Vault, the commercial tool that we've brought in. And we really did work with the user experience to see transitioning between tools. And I would imagine as we get this all together under the BCB portal, again, under a single umbrella, that exercise will go in again, where we interview a user base to make sure that the workflow and the website is designed so that the user experience is both beneficial to the research, but also friendly enough that people will come back and use it over and over again.
Sure, thank you.
Yeah, I think, Rob, what type of user do you target for this portal?
Primarily experimental biologists.
Oh, okay, got it.
Yeah, because our idea is like, because in our group we are most people in our group is like computational biologists, so that we can do the algorithms, we can do the analyze workflows, we can do the analysis by ourselves. Then we provide this kind of analyze results to the experimental biologists within this portal, so that they can directly use them.
Yeah, I think it is great, to see a lot of, statistics bar and the information. Yeah, pretty cool, Jian. Jiannan, right? Okay.
Yes, yes.
Yeah, thanks. Yeah.
Yeah, thank you for the comment.
Yeah.
So I'll just continue to quickly go through because there are a few things left in the system. So in the Toolbox AD part, we provide customizable analyze tools with integrated data because we have several, currently we have, I forgot how many we have, but we have several data sets, GEO data sets, the mRNA expression profiles integrated into the system so that by using those kind of data sets, we can run some customizable analysis. And we have 3 in-house tools here and some external ones. So the external ones just an external link so that you can go to their website to check out all the functions. But for the first 3 here, we have the first one is the ID converter. One common challenge for computational biologists is like when they're trying to identify gene lists and use the gene list into different tools. Different tools may require to use certain gene IDs. What I mean by gene IDs is like sometimes gene symbols, sometimes interest ID, sometimes ensemble IDs, those kind of stuff. So they have to just manually check them one by one. And then, but when your gene list is very long, I don't think there is a tool available to directly convert the gene IDs to other types. But here we provide us like a simple tool, a simple UI, which can be used to convert the gene IDs. Like if I have a gene symbol in my input, so I just select gene symbol, then I need these 3 different gene IDs, then I just copy. So this is the gene list I use for the gene ID. You can just convert and you will see here all the corresponding gene IDs will shown here. So if there is a gene that not identified in the system, then you will see it here. We do have some the recommendations based on your input. Then you can export it into a CSV file like this one. So this is a query I just used. And then you can see all the other corresponding IDs here. So that you can use it directly. This is the first tool. Second one is a PCA plot. This is kind of data exploratory tool, which PCA basically means a principal component analysis. So you can choose one of the data sets we have in the system, then do a PCA plot to see the variance in the gene expression profile. This is a basic check of the variance of the profile, whether you can use this data set for some downstream analysis. So the principal component analysis is usually the first step people will do when they try to use a certain data set. So here we just provide a function to explore the data. And also you can color code all the data points in the gene expression profile with different criteria, like here, the AD status. So we color coded all the points, the data points in the data set with the AD status, whether it's AD, whether it's HD is a Huntington disease, and whether it's a control. So you can see whether this kind of principal components can divide all the data points for certain attributes here. So you can plot these attribute different things. And you can also change the number of principal components. Also, we have a 3D visualization here so that you can check it, check the PCA plot in a 3D format. So that it's much more, so it can include one more principal component here so that you can check it. This is the second tool. And third one is the GSE, gene set enrichment analysis. So we have a user interface, like if you just choose one data set and you separate the patients with some clinical features, for example, we use this data set, and then this is a group 1 and this group 2. So this is like the control group and test group. So we want to see what kind of pathways are enriched in the control group, in the test group. So we will divide the patients into different groups. Like here, we can have like younger patients, 19. So this is the minimum, and then 45. And we apply this filter. So there are 33 patients within this kind of age range. And then there's no burn region information. We can just use all of them. And then we can set up the group 2, and maybe 40 to 20. I just randomly type in some numbers. so that we have more patients, then we can confirm and then go to the next step, trying to set up some parameters for the algorithms and then run the analysis. Because when you run the analysis, you can provide an email, or you can just option out with the email, like you can just choose agree and submit, and person, okay, yeah, and then you will have a analyze number, or not analyze number, so just ID, a unique ID for the analysis. Then we can yeah, so I just saw questions. So, no, not all of them, but some of, like for the GSE. So this is a good example of the task handling of the system. It's like the task will be submitted to the computer, and then it will run for some time. I'm not sure. Depends on the size of the data set. If you choose a larger data set, it will run like 10 minutes or 15 minutes. If you choose a smaller data set, it's like 5 minutes, 4 minutes. And with the unique ID, you can view the result. So it's okay, it's running right now. But when the analysis is done, then you can view the result. I think it should take like 5 minutes or so, but we can come back to check later.
And Kenneth, just to go on that, some of the stuff that we'll talk about next, we'll talk about some mass spectrometry based research. So some of the stuff under the BCB portal or the TREATAD does use batch computing, and I'll mention that briefly. Next. While Johnny's tool itself isn't using batch compute resources, some of the AD tools that we're using do.
Okay, yeah, and then we go through the TaRget AD. TaRget AD just basically integrated multiple resources for target evaluating. Let me use the gene list again here to demonstrate it, because you can have a gene list here, and then if you query the system, so there are some unidentified genes, because we don't have all the information for each of the genes available. So, but we do have like more than 10,000 genes information here. So if you have a gene list, then you query it, and then it will show here with some basic gene information. And then we have a spider plot with multiple dimensions, actually 10 dimensions. They are all from different, like some of them are from Farrell's database, and some of them are from Campbell. So when people are trying to compare different targets, like gene targets, they will evaluate whether this gene is a good target for the AD research. So they can just check this like kind of spider plot with multiple dimensions and trying to compare them, whether this target is good in some way or not good in some other ways. So we can compare them directly. So all the detailed data are shown in the table on the right. So you can just place your mouse here and try to see the specific like a definition of the attributes in the spider plot. And we also have a thing called the score indicator. So for some like float-based scores, we want to give the user a clear understanding whether this score is high or low comparing to all the genes available in our database. So the box plot is generated from all the available virus novelty in our database, and the certain genes like this one, the DIO2 or this one genes score is indicated with a red diamond here, so that you can see whether this score is truly high or low compared to all the other genes. Just a tool for it will be used for targeting evaluating. And the data AD is like it will be used for data export because we have all the clinical features available in the data set in the system, and then based on those clinical features, we can get part of the gene expression profile exported. Because like for example, just this GEO data set, we only, for some researchers, we only need like some like a more senior patients. Like we want to check the patients 70 years old or later or older. So it's like we can define a range and apply this filter. And oh yeah, this one has lots of different brain regions. So if we focus on some certain brain regions, I just randomly choose some. And we have 100 patients within this age range. And okay, let's just add one more. And we focus on the yeah, so there is something wrong, like the female patients, we just confirm. And so the female patients is like between this age range is filtered out. Like we have 78 patients, which after filtering with these two filters, and we choose the certain brain regions we are interested in, and then we can export a CSV file with all the, the gene expression profiles for these 78 patients. So this is just so it will take some time because the size is kind of big. Yes, here we go. So we have a CSV file with the corresponding patient's expression profile for all the genes here. So this is a kind of big file. So this is one is used for data manipulation. It's like some researchers may focus on some certain group of patients based on the clinical features. So that we provide this kind of tool to export the data with clinical features to export the gene expression profile. Yeah, this is basically all the modules in the ADE right now. Some expandable things I want to mention is the toolbox. So because we have the data infrastructure inside, in the back end of the system, so we can incorporate more and more tools here very easily by using this system. And another thing we can expand is like we do have a admin page. We can import expression profiles and together with clinical files, and then it will be directly shown in the Toolbox AD. So if you click one of them, you can see a list of available data sets. We can incorporate the data very easily. Then for other functions, you can check the documentation page. There are some brief introductions of each modules here. We also included a case study using online GSEA, and the case study we use is published study, and you can validate this kind of analysis result from the GSEA with the existing publications. Yeah, that's pretty much all.
Thanks, Johnny. That was a great description.
Yep.
Yeah, very
Thanks
good.
for listening. So it take a little bit longer than I expected, but there are lots of things to cover here.
So
Yeah.
Rob, you think you can wind up in like 5 to 7 minutes or so?
Yep, I'm gonna push through this, and hopefully I can leave about 10 minutes for questions. Again, I'll go through some of this really quickly. Let me get my presentation back up here. So as I was saying, based on the question earlier, there are other tools here within the BCB portal. One of them that actually has its own gateway now is the Topics. This does mass spectroscopy for proteoform identification and characterization. There are 4 MS tools here you can see listed, and this is a standard Apache area of audit interface, and also a conversion tool here for file conversion. It uses currently local resources here at Indiana University, but it also uses UCSD resources. So it's utilizing the Exceed infrastructure. Another tool that we've been involved with, and for some reason the arrow keys aren't working, is the Tsunami tool. This is a translational bioinformatics tool suite for network analysis and mining. I put the workflow in here. I won't go over this based on time, but if anybody is interested here, this is at biolearns.medicine.edu. You can explore that tool. There's also a tutorial which is being created. The tutorial button now is empty, but there is one being created. I won't talk about this at length, but inside of this project, something we didn't expect that happened in year 1 is really the medicinal chemists and the structural biologists really wanted this CDD Vault, which is a commercially provided cloud resource. So there's some integration of not only the BCB systems, but data from a cloud vendor CDD Vault that will be integrated into this. There's been a lot of additional complexities. We ended up, because we saw the complexity going back to start and creating a data management plan. This was not only necessary for the project to understand the data, but it was required by IU in our procurement system to have a data management plan before we purchased the CDD Vault licensing. Again, I won't go over a whole lot of this, but we are looking at operating this for many different reasons, and the data management plan helped out with things like onboarding and offboarding procedures, sponsor and sharing requirements, quality assurance, quality control. So the data management plan actually helped us a lot in the operational aspects of what will eventually be a unified portal. Let's see, we took away several things from year one. The project became much more complex, as I mentioned. There was a lot of general IT support that was needed here. Also, a lot of things like domain names being created and registered with DNS, just some general help with hosting on Jetstream, and creating just standard web servers. We're still early in the process, year 2 of 5 in this project. So as I said, not only do we understand the complexity a little bit more, we are now working on plans, and there's been actually some change in things. As a program officer, would like to see the data portal, the public data portal, be a collaboration between Emory and Sage BioNetworks, along with IU and Purdue. So basically not present the same data in 2 different interfaces, or similar data in 2 different interfaces, to avoid confusion by the AD research community. We're working with Johnny to move his stuff from where it's hosted now on an individual server to a area of out portal. This will help with operations long term. It also help with future development that may be done by another developer, as Johnny is getting very close to graduating. I think maybe at the end of the spring semester, Johnny is done. So we'll need to have these AD Explorer tools that he just demoed at a place where they can be operationally maintained, along with new development being done, and not basically run into the lack of additional development after Johnny graduates. We're revisiting the workflow between the structural biology, medicinal chemistry, and the high throughput screening group. One of the things that we've been told over and over is that the structural biologists and the medicinal chemists speak a different language, and what they'd like to try to find is some format that when they find a target, that both can easily understand the promising aspects and the reverse aspects of a target. Just because the medicinal chemistry sees promise in a drug, that doesn't necessarily mean that the biologists will, and communicating amongst those groups is important. And can we provide a format for documentation where everyone can quickly determine whether a drug should be used as a target or can be discarded for some reason. The initial prototype is at bcbportal.medicine.iu.edu. This pulls together the PubAD tool, one of Johnny's tools, along with the topics and the tsunami under a single resource. Eventually, we're hoping that all of the various different tools that are developed will live under this bcbportal.medicine.iu.edu and be a AD research portal for anyone who wants to use the variety of tools that are being developed, at least within the IU School of Medicine and Purdue groups. And then on my end, we want to apply fair data principles to anything that we publish to be sure that it is reusable at some point in the future. And I'm going to wrap up there. And I went through these last slides very quickly, so but I do want to leave a little time for questions, so we don't leave you with questions and not answered.
Thank you very much, Rob and Johnny. This was a fascinating presentation about like an amazing and, may I say, very ambitious project. And I guess, as you said in your last slides, you're starting to discover exactly how ambitious. So hopefully it'll all come together. First off, I see two questions here in the unanswered questions here in the chat. So one from Ken Yoshimoto. Do any of these tools require batch compute resources, or do they all run immediately?
So and I think we covered this one a little bit, but the tools within the AD Explorer currently are all running locally. However, the things like Topics, Tsunami, and other tools do use batch resources on other Exceed resources and local IU resources. So depending on the tool and its computational needs, batch resources are used, but not by all tools.
So if I may, I mean, that kind of raises a corollary. So to what extent is the project dependent on access to what kind of, computational data resources? Is that like covered in the, and by the way, did you say this is NSF or NIH?
NIH.
NIH, okay. So to what extent is, support for the back end, like the compute and data resources, including in the grant, or is that something that you guys have to somehow scrounge, or what is that?
So right now, the idea is to use either local resources or nationally provided allocated resources. Again, at least the topic gateway uses UCSD and IU resources. Though really it's going to depend on the tools. So I think even though we are using national tools right now, it's not at the size that it really needs to use UCSD. It could run locally here at IU. However, we want to leave that open, and we want to do that integration. One, because it's already done within the area Vada environment, but two, mass spectroscopy can be very demanding in the future. And in fact, I think a few people on this call probably saw me asking for letter support for a mass spectroscopy project that we are submitting tomorrow, doing proteomics. So MS is potentially very, very heavyweight, and we would need external resources for that.
Okay, and then the other question here is from Chunhan. When exporting CSV files, do you follow some standard format?
I think the question is for me. So the CSV files we are exporting is just a gene expression profile. Each column is a sample, and each row is a gene. This is pretty common format for the gene expression profiles. So it's very straightforward, I think.
And Chunhan, I think you have a follow-up question, which, if you don't mind, just ask it yourself.
Yeah, I think some like a CSV file can be used to analyze, in this data in some other tools, something scientists using some specific tools, and then load up. That's what I meant. My asking is, when you export any data, just like you can have some a common or standard format in biology or history or something, yeah, that is my questions.
Yeah, and Chunhan, thanks for asking that question, because that gets to that very final bullet point in the FAIR data, right? So is there a definition of the format for the CVS file? In fact, I would like to see this registered as a data type. So when somebody gets the CVS format, they can actually go to a data type registry and determine the data type, not only for each of the individual columns, but for the overall file. So I think fairness actually requires something along those lines, though. I think now what we're using is just kind of, as Johnny said, just kind of the standard delivery that we know the researchers that we're involved with are using. If you want that to be used wider, especially by an international audience, then you really need to have a definition of all of the aspects of the data that you're delivering, whether that's a CVS file or CSV file or anything else.
And Jiannan, you also had a question there about the framework for data sharing, I guess. What kind of apps?
Yeah. If you serve any data to users, Rob, Jiannan, I especially interested in how you guys creating data sharing system in your portal, So just access local disk or peer S3 or Globus or some other things. I know about you guys have some plan for that, I think.
Yeah, so this is the thing that changed between year 1 and year 2 based on a program officer's request. We were going to do the data management and data delivery through the Aryavata interface. However, the program manager said, well, don't publish 2 similar things in 2 separate locations and get the community confused and give them, basically information that can be misunderstood in some way or preferenced one over the other. So what they've asked us to do is actually to create these target enablement packages and deliver them to the Sage BioNetwork system along with the metadata that they need for reading that. So we're not going to serve the target enablement packages directly anymore based on feedback from the program officer. We're going to actually make an interface for Sage BioNetworks to get the target enablement packages and then publish them through the Angora, there's another name, interface. So now it's gone from where we directly publish to where we're sending it to somebody else to publish. Though there are not all these tools are part of the target enablement package. So the stuff that Johnny's doing and the stuff in topics, we will publish that if there's not a related tool within the other TREATAD group.
Just real quick, what's the size of those packages you're going to be sending over to Sage? Are they large or tiny?
Yeah, they're not large. So the data is compiled into a PDF file that is kind of like a has some visualization and some text in it, but it's not a huge amount of data. It's going to be basically a nicely packaged conglomeration of all of the other stuff that happened. So we're not sending the data, we're sending basically the summary along with visualizations and the analysis work that we've done and some explanation of that.
Yeah, Bob, actually Kenneth and I, also have a similar, not similar, brain science data, project that the UCSD, that's why, we need a little bit of what you're gonna try to do, Well, thanks, Rob. It's
And I mean, one of the reasons why we have these ECSS symposia is so people can figure out, hear what everybody's doing and then continue the discussions and stay in touch. If I'm sure there's more conversation that you guys want to have. And even here at PSC, we have some kind of similar in scope projects. And so I think all of us probably at some point will want to talk some more and learn from each other's experiences. But since we're 1 minute over, I would like to—
Sergio, just let me say, feel free to reach out to me. Jiannan, I know you know how to reach me, and anybody else who's interested and has similar things. I can say that we've submitted a NIH proposal for a similar type of portal for cancer research. So we also think that this has application, or at least this type of pulling tools together has application in other research areas.
Tools and data.
Yep.
Yeah. Thanks, Rob.
Thank you. So thank you very much again, Rob and Johnny. Marcus, I guess maybe in early January, this recording will be available on the CSS Symposium website.
Yep, that's correct.
So thank you everybody for taking time out of the holiday preparations to attend this. I think it was well worth it. And thank you everybody for all your great work in 2021. And let's hope it's going to be a successful 20 and safe and whatever 2022 for all of us. So thank you very much. Happy holidays and see you all next year.
Bye.
Thank you. Bye-bye.